So what's going on, guys? My name's Bryce, and I'm here for the Nanoscience program. This is a niche, and this is Matt. We're very close, and today we are presenting to you our what we've learned here, pretty much. The specific topic today is plasmonics, specifically plasmonic resonance. And in order to understand that, you need to understand a little bit about nanoparticles. We've already had a group go up and talk about plasmonics, so this might just seem like a blatant ripoff of Nora and Becca and them, but I swear we came up with all this on our own. So nanoparticles, the ones that we've created at least, we made in about 2 hours. So they're a pretty simple thing to make. The word like nano and particles, you think of like some super sciencey thing, but they've been around ever since like the ancient Romans. They had that cup that changed colors. Notre Dame is old. The rose window, that. Is it not old? It's an old church, and the window there, the light, the change of color is all due to nanoparticles. So despite the fact that people haven't known about how nanoparticles work, how to create them and whatnot, hasn't changed the fact that they've been useful, and they're going to continue being useful for all time. So how we created nanoparticles was we used a metal. And for nanoparticles, you want to use a Nobel metal. I don't know if people here are familiar with the periodic table, but Nobel metals are more stable than other metals. And so like gold, silver, copper, that's what you want to use for creating nanoparticles. And in addition to that, you need what's called a capping agent and a reducing agent. In our experiment, citrate was both of those things. It was a better reducing agent than it was a capping agent. And so to explain capping in basic terms, because I don't understand it in complicated terms, it basically keeps all the nanoparticles from grouping up and aggregating. That's a word that was thrown around a lot. And reducing agents, what they do is they give away electrons, which is kind of counterintuitive to the name, but they give away electrons. And the only thing else that we had to do in the reaction was stir it and heat it up. And heating it up was just to speed up the reaction, as was previously talked about. And so it's a pretty simple process to create. The nanoparticles that we made were called nanostars, and this is something that Anish and Matt will both talk about as well. But the reason why nanostars are interesting is because they have a higher surface area to volume ratio due to the fact that, like a star is pointy, it's got things coming out of it, so there's more surface area to that. So that makes it more unstable. So that's something interesting, and just keep that in mind as we go through the presentation.
All right, so on the surface of this analogy, it's obvious that there's an opera singer reaching an extremely high note, as you can see, and that frequency and sound being absorbed by not the smaller wine glass, not the bigger wine glass, but the one particularly in the middle. And then in the next frame, you can see it shattering because of how strong the frequency of the opera singer is. But this is meant to manifest a big concept in nanotechnology of the scattering of light and the absorption of light and the scattering of light in that order. So what's happening here is that when light hits a nanoparticle, for it to appear to like the eye, the frequency of the light has to be the same as the frequency of the electrons that are oscillating. So that would mean that, and it particularly hits the middle glass because those electrons are oscillating at the same frequency of the light, while the bigger particle that's displayed in the bigger glass, the electrons are oscillating really slowly because it's slower than the frequency of the light because the particle is bigger, and vice versa with the smaller one. And in the next frame, it is shown that middle glass completely shatters, it explodes, and with all those energy beams and stuff. I don't know. And so what that is intended to represent is the scattering of light, and I think that a limitation of this is the fact that a color plays a big factor into this, because the color of the light absorbed is actually complementary to the color of the light that is scattered by the nanoparticle, and yeah.
All right. Well, beyond analogies and simply making gold nanoparticles themselves, there are actually a lot of applications, especially in the biomedical field, for nanoparticles. One effect that was kind of touched on by Bryce earlier was the photothermal effect. And so in these gold nanostars, because they have this higher surface area to volume ratio, they're able to collect heat much faster than other shapes. So, for example, when targeting tumor cells, one thing you could do is attach these nanostars to the cell, and then you can use infrared light to actually heat up and destroy the cell. Additionally, if you wanted to use nanostars differently, you can directly target and deliver drugs in a more efficient manner so that, say you have cancer, you wouldn't have to undergo chemotherapy. Additionally, you can treat allergies using nanoparticles. So by using these nanoparticles, you can target the liver, and what that would do is suppress the immune system so that in case you eat anything you were allergic to, you would no longer have that allergic reaction. You can also have this opposite effect where it would excite the immune system, in which case you could help fight off any illnesses. Outside of the biomedical field, it was mentioned in the first presentation, but with the rose window, you can make stained glass by choosing differently shaped nanoparticles, which in turn produce different colors. Thank you.
Yeah, thanks for
Thank you.
listening.
Questions?
Nice.
Taught them everything. They don't have any questions.
Yeah.
Right. So you mentioned at the end targeting the nanoparticles, so getting them to go to the tumor cell or to the liver for allergies. How exactly do you target the cell? How do you get it to go where you want it so you can heat it or so that it can prevent the allergic reaction?
So generally, you can shape the nanoparticle very specifically, depending on the shape, size, the material, and the environment. And so what it does is it's structured so that it only attaches itself to that specific cell you're targeting.
So I'm going to add on to her question to give you guys a little bit more of a chance to answer. So the shape and the size doesn't necessarily dictate where it's going to go. In some cases it can, but more specifically, there is a thing that you can put on the nanoparticle to get it to go where you want it to go. So when you're synthesizing nanoparticles, you mentioned something that prevents it from aggregating, which is
Capping agent.
the capping agent, right? So you can coat nanoparticles with things. So there's a thing that you can coat the nanoparticle with that will make it attach to or go into specific things. Do you remember the term for that?
Antibody? Oh, it's an antibody.
It's an antibody, right? So you can attach molecules, just to answer your question before, you can attach molecules so that's why it would go to the cancer cell specifically, because the cancer cells would respond to it, whereas other cells in the body do not.
So is it live or is it Memorex?
Sorry, what was that?
That was a joke. That was meant for the older crowd.
Oh, sorry. Yeah, no, I'm not in the older crowd.
Oh, you're a boomer.
I'm a boomer, yeah.
Seems like it's live singing to me, but it could be Memorex, I don't know.
I don't get it.
I'd like to think that the artist was depicting me singing because of the curly hair, but who's to say? I don't know.
That's a really high note, too.
Yeah, I mean, I'm no soprano, I'm an alto, but, I try. Other questions?
So you talked about the difference between stars and like regular spheres. How is the photothermal effect different in stars versus spheres?
So the nanostar has a greater surface area than the sphere. And so because it's more spread out, it can absorb heat on a greater amount of surface, and then it can heat up quicker.
Specifically, where in the star and where in the sphere would you have hotspots?
Specifically,
It would be the tails, right?
yeah.
Or the tail ends of the stars, or the points, yeah.
Yeah, so the photothermal effect is much higher there, right?
Yeah, and that's like what makes it more unstable.
So like Bryce stated earlier, kind of like with increasing instability or like a greater surface area to volume ratio, you can gain heat quicker.
I guess the last question, another question if no one has hotspots.
I have a question. So you talked about being able to, tune nanoparticles based on size and shape and get different colors as a result. There's a really big application other than medical applications that specifically utilize the fact that you can produce these colors. So do you know what that would be?
Is it Raman spectroscopy?
That's a
That was-
way of being able to analyze stuff. But I mean, like, so there's a big actually consumer electronics application that uses the fact that you can generate a lot of colors with these particles by exciting them.
Would these be screens, like
Yeah.
television screens?
Yes.
It's like a cathode.
So can you describe maybe a little bit about how that might work? So how would you, so you would make different colors based off of what? Changing three, you can change three things, or you can
yes.
change three parameters to make
So
different colors, right?
you'd have green, red, and blue lights, right? Or
Well,
that's what they have in-
so you can change the color that the nanoparticle is based off of changing what things? The size, the shape,
composition.
and the
Temperature.
composition, right? So when you're using nanoparticles specifically for the color or to, and this is, you saw this in the light microscopy lab, right? Where he used these different nanoparticles basically as like staining tools to like color stuff. Do you remember
Yeah,
what those were called?
Wait, he talked a lot about quantum dots. I don't remember.
Yep.
Oh, is that what they were called?
Yep.
Oh, no way.
Wow. Okay.
So quantum dots are actually a really big application right now for television screens, right? So if you hear about like QLEDs, those are trying to use these plasmonic quantum dots for television displays. Other questions? Last question? And if, oh, yes?
Wondering more about the experiment. What colors did you observe, and were you able to extrapolate what kind of shapes you were creating?
So how we figured out, or I'll start with the color. So when you use gold, as you can note in the rose window, it creates like a red pinkish color, right? And we learned a lot about complementary colors. So say you reflect something that is purple, you get
Yellow.
I was about to say that. You get yellow as the light that comes off it and the light that you actually see. In terms of how we actually know that we created the stars,
Well,
how do we know that?
There's
we
a
made-
they were
change in color.
they were unstable, and when we were heating them up, they were changing colors. If you were to stir the water, it went from like blue to like purple.
But like, how would we tell that it's a star versus a sphere?
Because the spheres are more stable than the stars.
Well,
said.
you also used a technique in the afternoon.
One thing is a spectrophotometer. And so what it does is it measures the absorbance of light and which frequencies of light are observed. And so the way it works is it shoots a beam of all these different frequencies, and it picks up the absorption at the back. And so when you see two peaks on the graph where two different types of absorptions or wavelengths are absorbed the most, you can see that there are two different colors being absorbed. And the way, when you see electrons oscillate inside the star, they're kind of going on different planes and at different speeds, where on the point of the star, they're going much quicker. And so they're going to produce wavelengths of higher frequency and a different color. And in the sphere, they're going to go much slower, so that they produce light of, like, say, red.
And then what was one last way that you saw the different shapes, or that you knew that you had the different shapes? What technique did you do to actually verify what your particles looked like?
Can I get
You stuck them in a big machine that let you observe what they were. Do you remember what?
microscope?
Mm-hmm.
Oh, okay. Come on.
So what's the resolution of an electron microscope, roughly? What is it?
I thought you said it was an electron microscope. My bad.
Sorry, I'm asking, what is the resolution of an electron microscope?
Okay, so electron microscope. I'm not sure about like an exact, like if you're looking for like a quantitative.
Like
Roughly-
nanometers.
roughly-
A few nanometers.
A few nanometers, and that's because how an electron microscope works is that it actually shoots down like an electron beam, and so like things like air, dust, and whatnot can mess that up, and so you're looking at a very
Airtight.
Yeah, a vacuum. You're looking at a very small area, and then as we also learned in, I forget the name of the lab, but you can basically wrap a laser in another laser and then squeeze it super tight, and theoretically you can create a viewing, I don't know, like a viewing size, I guess, of like less than 1 nanometer, but due to the fact that you can't actually create a laser that squeezes it that tight, it's theoretical, it's on paper, it's possible, but in real life, it's not practical.
Any last questions? All right. Thank you, guys.
Yeah, thank you.
Thank you.